Hello learners, today's topic of discussion is quality control. So what is quality control? To understand that term, we need to know what do you mean by quality. Since I have pharmaceutical background, I'll discuss about a drug and what quality we seek in a particular drug. So whenever we are talking about a drug, what we think about that a drug should be of a genuine quality and of good nature. A drug should be physically and chemically pure. A drug should contain the same amount of ingredient as stated on the label. A drug should be rendered in such a form that it is effective after administration. A drug should retain quality in terms of shelf-life or stability. A drug should not have toxic impurities which render it practically useless. A drug should not contain undesirable impurities because they may produce undesirable changes during dispensing or processing in the laboratory or industry. So, if this is the quality, what all things we need to do to attain this quality? So, what all measures we take to maintain the quality of drug is known as quality control. So, the term quality control has achieved much importance in pharmaceutical industry. It is almost essential that a good quality product should be available to the doctors for treating patients or for the actual users. Thus, the responsibility of pharmacists and those of pharmaceutical industry increases appreciably, so that good quality pharmaceutical products should be available in the market. So, the term quality as applied to drugs and drug products is said to include all those factors which are contributing directly or indirectly to the purity, safety, effectiveness, and reliability of the pharmaceutical product. In order to attain the above referred properties in a drug, it becomes essential to undertake a quality control. However, it will be appreciated if one achieves a concept of total quality control in place of quality control. By the total quality control, it means to include all those aspects which start with the procurement of raw material to the finished product available in the medical store and until it gets consumed by the patient. Thus, it includes not only the parameters of good manufacturing practice but also the storage, handling, and preserving the sample until it is ultimately consumed. Also, a drug administered to patient should not cause undesirable reaction due to impurities. In order to maintain the discussed qualities of drugs, a quality control is essential in the pharmaceutical industry. The essential job of quality control is to test a drug for quality and quantity. In order to achieve both these parameters of qualitative identification and quantitative determination, the procedures and standards laid down by various national and international agencies are to be followed. In every country, the pharmacopoeia is the official compilation of standards of quality for all currently used medicinal agents together with methods used for such quality control. Furthermore, revisions are made and published periodically in the form of supplements to keep the pharmacopoeia up to date with respect to matters pertaining to the drug quality. One of the main areas of quality control is to carry out the analysis of drugs for quality and quantity. Various tests and procedures for analysis, including finding and determining impurities, are included in the official book. This is applicable both to the drug as raw material as well as in the form of finished product. The pharmacopoeial monographs provide detail about all this. In pharmacopoeia, every test or criteria prescribed as a standard is a parameter. Assay is another very important parameter. Solubility is another parameter. Limit test for arsenic is another parameter. More the number of parameters prescribed, more rigid the standards and more reliable the substance will be. In practice, however, too many parameters are not necessary. A few important and critical parameters ensures good quality. Also, too many parameters give rise to cumbersome and tedious analytical process and increase of cost of the substance. Hence, few critical tests are selected and prescribed by the pharmacopoeia. All the prescribed tests and procedures for a given medicinal agent or pharmaceuticals are given to ensure its quality. Thus, the scheme is termed as quality control. In the quantitative analysis, depending upon the characteristic of drug and its formulation, various analytical methods have been prescribed. These include physiochemical methods, whose principles are based upon some specific physical or chemical properties of a substance being analyzed. This will involve determination of specific gravity, density, viscosity, surface tension, refractive index, optical rotation, etc., and all the types of volumetric analysis. While utilizing the physiochemical properties, use of instrumental methods such as pH potentiometry, conductometry, polarography, colorimetry, spectrophotometry, fluorimetry, flame photometry, etc. have been recommended. The other methods for quantitative determination include the separational techniques such as chromatographic, gravimetric, precipitation, and complex formation, proximate assay, and special methods which need distinct type of arbiters such as volatile oil determination, alcohol determination, techniques of oxygen flask method, etc. Besides the usual qualitative and quantitative determinations, pharmaceutical products have to be evaluated for their quality. For instance, the studies and tests like disintegration, hardness, friability, dissolution for tablets and related preparations, bioavailability studies, absorption, elimination studies, pharmacokinetic studies, biological and microbiological studies have also to be carried out. The basis of all the studies and tests of quality control has been to ensure that quality drug becomes available to the consumer and patient. So, what all steps and what all things we should perform while doing this type of activity or performing the quality control of our pharmaceutical products have been classified into three categories. That is, first one, perform following activities in routine manner. Second, activities managed through and non-routine. So first one, in routine manner, we have to continually and regularly we have to do such activities like we have to analyze the raw material and product manufactured on daily basis, that is on time to time basis, not only daily basis, time to time basis, that is when the production starts and as soon as it finishes, we have to check or analyze the quality of the product along with the raw material. We have to, in between, we have to also check the intermediate form. That is, if you are doing for tablet production, then you have to also check the granules, that how it is of good quality or not. Likewise, we have to go for various dosage forms which we are preparing. Then we have to go for the finished good analysis, that whatever product you are targeting, when it is being completely produced, you have to go for the analysis of that finished product too. And then when you have analyzed the finished product, you have to also check whenever this finished product is being kept for some period before being consumed, that should be long enough. For determining such life, that is called shelf life of the product which you have produced, stability studies are being performed so that a suitable lag of time can be given as a shelf life to your produced product. So, what all activities you need to do or you need to perform for getting all the analysis done for your raw material, manufactured product and intermediate product. What all things you need to do and what all activities are being done so that their quality could be maintained and their analysis could be done include instrumental analysis, chemical analysis, microbiological analysis and packaging material analysis, and this analysis can be done by the methods which we have stated before. Non-routine activities which you need to follow so that continually a good quality product can be produced in your industry includes the calibration and preventive maintenance of the instrument, preparation of reference working standards, and method development and validation. These are non-routine that you have to not perform all these activities on daily basis, but that should be done on regular basis at fixed interval of time, so that all the instruments which you are using for analysis and for production and whatever other included activities being done need to be done for carrying out a production of good quality. We need to go for these non-routine steps, non-routine activities on regular basis too. So, what all components of quality control are that could be understood and that can be achieved by following points, that is, adequate facilities should be there so that you can maintain quality control, suitable quality control. Trained personnel should be available so that he can carry out this quality analysis in a better way so that you can achieve the quality control. Approved procedures must be available for sampling, inspecting and testing of starting material, packaging material, intermediate bulk and finished product. Wherever appropriate monitoring is required, then we need to monitor the environmental conditions also so that microbial contaminations and other type of environmental contaminants could not get incorporated in our product. So, this is how we can maintain a good manufacturing practice, and that can be achieved, which is the main goal and main aim of industry to achieve GMP process, good manufacturing process, that is called. So, what all components of quality control? So that what all things we should keep in mind so that we can achieve a better quality control in our industry while producing includes records. Most important thing, that is records. Whatever you do, whatever things you do, that should be recorded. That too, in written format, that should be available so that if anyone wants to cross-check or recheck your data, if any deformities or any complaint comes around or any type of inspection is going on, so that they can check that you are maintaining good quality control parameters in your industry, then what all things they need to know, that should be properly recorded in your records, and you should maintain records of each and everything which you have done to maintain the quality of the product. Sampling, proper methods, proper utensils and proper gears should be available, and those all things should be properly defined, that what for and for which type of product you are going for. Like if you are testing taking samples of raw material, or you are taking samples of any intermediate product, or you are taking samples of the packaged goods, or you are collecting samples from the retail shops from where you have delivered your product, manufactured product, from that place if you are collecting. So what all methods of sampling should be there, how the sampling should be done, and how much amount of sample should be collected so that proper results could be generated and we can check that quality control has been maintained or not. So sampling is a greater area, has a lot of importance. Next one is quality control labs. To maintain better quality control, we need to have good labs. That is, a lab having good facilities, having good instruments, good number of instruments with good quality instruments, so that proper quality parameters could be checked and the quality, the ultimate required quality of your product could be maintained. Next is validation. Validation is, you can understand this thing right now as a well-structured process. Validation is a well-structured process by which you get a good quality product if you follow each and every step. Then such process which guarantees the production of a good quality product each and every time is termed as validated process. And when we check and recheck any process of production and we finally say that this is how, what all steps you need to take so that you can get a good quality product, then this type of process which is being done to assure that this process of manufacturing is good enough is called as validation. Okay, to generate a validated process, you need to do validation of any manufacturing process. So validation is quite essential so that you can get or you can assure that whatever you do will yield a good quality product. Next one is batch inspection and sampling. Though you have a validated process of manufacturing, but then also you need to keep an eye on each and every batch which is going produced in your industry, so that by any mistake, by any reason, if there are minute chances or added chances of spoiling your product, that should also get checked, and that can be checked if you are monitoring each and every batch and taking proper samples from each and every step of your production. This will further guarantee that by any means no chance is there that your quality will be hampered in any situation. Next one is retaining the sample, retain sample. This is done so that whenever any reports come from the consumer side that after producing you have sent your product to the market, and from there you are getting any complaint about your product. So to cross-refer those samples which are available in the market and your batch which you have produced, to cross-refer those two samples, few of the samples should be retained in the industry so that you can, first thing, you can get safeguard from the people who will show some low quality product of some other industry or some other manufacturing unit as your product. Then you can easily claim that no, this is not our product. Yes, our product is having all these qualities and this is the sample of that batch we have delivered to the consumers or the retailers or wholesalers, and whatever sample the individual is showing is not your product. To justify that point, retaining the samples could also prove beneficial. Now, analysis of finished product. Whatever product you are manufacturing, when it has been completely manufactured and ready to be delivered to the consumers, for the consumers, you need to further go for analysis of those finished products so that you can confirm or ascertain that you have produced a good quality product and now it is ready to be consumed, and so we can deliver it to the market. And this is how, if we maintain all these points, we can achieve quality control in a better way. So, what all things should be followed in a quality control lab, and what all points you should keep in mind when you are in a quality control lab are here under. That SOPs, that is standard operating procedures, should be there for each and every instrument, so that any individual who is using any type of instrument for analysis and quality control checks can use those instruments in proper way and get correct results, so that the quality of your product can be maintained throughout the process of manufacturing till the completion or the finished product you have produced. So, SOPs of each and every instrument should be there, and those SOPs should have validated procedures so that an individual can contribute in a better way to the quality control of your product being manufactured. Documentation room. There should be a room or facility in your premises of your industry where all the documentation could be done, and those records could be kept safely so that whenever anyone wants to check that proper quality control parameters have been followed or not, each and every step has been done or not, or each and every step is going in proper order, that could also be checked. So for that, documentation is essential, and for that to carry on those documentation process, there should be a proper facility. For that, documentation room is essential. Next one is instrumental analysis lab. These labs, they are the strength. This is the actual strength of quality control labs. Higher sophisticated instruments, if you have, proper and accurate result will be there. If the advanced instruments you are using, then you will get the results in short time, and that too in very accurate form. So the instrumental analysis labs should have advanced instruments and better facilities so that you could get good results of your analysis being performed in that instrument lab or your quality control lab, and you could actually work in a way so that a good quality product would be produced. So, instrumental analysis lab should be there with advanced facilities. Microbiology and toxicology labs, they are also essential, since due from environment, various microbes could get incorporated in your product, which will hamper the quality of your product. If not at the time of production, maybe when it is being stored in the storehouses or warehouses, or it is being kept with the consumer for later on use. And if such thing happens, this will again put a bad mark on the industry. So, such type of contaminations which could get incorporated, like microbiological contamination or toxic substance contamination, those type of contamination should be checked, and that can be checked if we have this suitable facilities available in our industry. So quality control labs should also have microbiology and toxicology labs. Chemical testing labs, these are very essential. If you don't have microbiology lab for very small scale industries, that is quite acceptable. They can send their samples to another higher labs for microbiological and toxicological studies. But chemical test labs are very, very essential, because this gives good results, and they are very much helpful in maintaining the quality. And these are very easy steps and easy procedures are there in chemistry to check various quality parameters of any pharmaceutical product. So chemical testing labs, these are very good labs, and they are very important, and they can be set up in a very cheap cost, though few of the chemicals could be costly, but these are very easy settable labs, and these are very much essential labs that should be there in every industry. Trained personnel, yes, you have all the gears and gadgets, but you don't have a proper person to work on those gadgets and gears, then again those instruments and gadgets which you have procured will go waste. So trained personals is utmost required thing for your quality control labs. So trained personals, they have good efficiency to work with the instruments. They have proper knowledge to overcome with the problems, if any encountered. They can even modify or change some of the chemical, physical, or any instrumental methods, so that proper analytical results could be obtained, and those can be used for maintaining proper quality control of the manufactured product or product under manufacturing. Provisions for retained samples and stability samples, like there should be every pharmaceutical industry should have provisions that how much samples they should keep with themselves, and how they should collect the samples from market, and where all they should keep those collected samples so as to be available whenever they are required for analysis. So proper provisions should be there for the quantity, quality, and storage premises for the samples which are being collected or which have been retained for after manufacture. So we are talking about samples from quite a long time. So how the sampling should be done? This is a good question that should strike in your mind. So for sampling, there are approved methods which needs to be followed while we are going for the sampling of our product or raw material or any intermediate product, which while carrying out the quality control checks in the process of product manufacturing. So approved methods for sampling should be there for the personnel so that they can follow the proper sample collection, and well-trained personnel and sampling tool suitable for as per the requirement of the product which needs to be sampled should be there. So a well-trained personnel should be there, and good tools for collecting the sample should be available so that we could not contaminate the product while collecting the sample. And record maintenance for all the sampling, that how much sample you have collected and from which process you have collected the sample, that all should be recorded and maintained. Checklist for every stage, that in each and every step of your manufacturing you have collected the sample, you have gone for the testing of those samples. So proper checklist should be maintained so that you could not skip any step. SOPs, another requirement, that is SOPs divided into and before, during and after. The proper SOPs for collection of the samples, that is before the manufacturing starts for raw materials, during the manufacturing is going on like for intermediate products, and after the completion of your manufacturing, that is for finished products. Proper SOPs should be available for the trained personnel so that we could collect sample on time to time basis, and those samples could be analyzed so that good quality product could be manufactured from the pharmaceutical industry. So there should be SOPs for your raw materials, the SOPs for finished material, and also there should be SOPs for intermediate materials so that proper samples could be collected from all these three types of products. So this is how sampling could be done, and sampling is very essential for maintaining the quality in the production or manufacturing process. Validation. All the methods used by quality control, say for sampling, testing, and other activities, must be validated. Validation of testing methods is particularly very important. I have defined for validation priorly also. So you can understand that validation is quite essential for maintaining good quality of the product in any industry or manufacturing unit. Records, we have discussed it prior to, that records must be made demonstrating that all the required sampling, inspecting, testing procedures have been actually carried out and that any deviation has been recorded and investigated. This helps in tracing the history of a batch produced. This may be required in future if any investigation is to be made. The basic concept of good manufacturing process is do as you have written, that is SOP, and write what you have done, that is the records. Finished product. The finished product must contain ingredients complying with qualitative and quantitative composition of the product described in the marketing authorization. The ingredients must be of required purity in their proper containers and correctly labeled. So this is all we should take care or we should have in our finished product. Written samples. Whatever samples we have written, that sufficient number of samples, sufficient amount of samples of starting material and product which has been obtained at the end of your manufacturing process should be retained and should be kept, and proper procedure should be followed to permit further examination of the product if necessary. The written sample must be kept in the final pack unless the pack is exceptionally large. So, the activities of quality department have been summarized here. First, they establish, validate and implement all the quality control procedures, maintain sufficient standards and reagents. Second, evaluate, maintain and store references and working standards for substances. Third, they ensure the correct labeling of containers, materials and products. Fourth, assess production, participate in the conditions. Fifth, participate in the investigation of complaints related to quality of the product. Sixth, participate in the environment monitoring results. Seventh, assess the finished product after evaluation. Assess manufacturing and packaging documentation. And the last one, compliance with specification of the finished product and examination of final pack. So now we conclude this lecture by the final remarks. In the final remarks, modern methods of manufacture of chemicals provide drugs of very high purity. In some cases, such purity can be achieved at relatively reasonable cost. In many cases, however, the purification cost may become prohibitively high. In order to keep the cost of medicaments at low level, some compromise has to be achieved in respect of impurities. This is a major consideration while prescribing standards. Again, this relaxation of standards of purity cannot become too liberal, both from an ethical point of view and from the point of view of the patient's well-being. The impurities should not be in a quantity that may be able to harm the patient or the process of manufacture, storage, stability, etc. This implies even if a substance is sufficiently pure to produce the required biological effect, the small amount of impurity may be harmful or undesirable and therefore is to be controlled suitably. So, this is how we conclude our discussion here. Thank you all for watching. If you like this video, do subscribe, like and share, and comment what you want to see next. Thank you all. Bye-bye.